Hello, everyone. Thank you so much for coming to the ADAPT Proposers Day. We're so excited to present the ADAPT program to you. My name is Andrea Bild, and I am the program manager for ADAPT, which stands for Advanced Analysis for Precision Cancer Therapy. We will start the day with Jen Roberts, Director of the Resilient Systems Office at ARPA-H. Dr. Roberts will introduce ARPA-H. I will then next discuss the ADAPT program. And then Caitlin Burns will discuss the submission process. And lastly, we will close with some important reminders. Thank you so much for coming. And Dr. Roberts, please take it away.
Thank you, Andrea. Hello, everyone. So I'd like to start off by talking a little bit about ARPA-H. Our mission is to accelerate better health outcomes for everyone, and I'd like to spend a couple minutes just unpacking what that means. So there's a couple different pieces to our mission. One is the accelerate piece, which speaks to the fact that our organization is particularly interested in revolutionary research that changes our perception of what's possible. We want to invest in technology breakthroughs and biomedical breakthroughs that can really help move the needle significantly in a relatively short period of time. Then we're also interested in better health outcomes, which means that while as scientists, we're of course very excited by basic research and the wonderful understanding that gives us. At the end of the day, what ARPA-H is interested in doing is taking those insights from the scientific community and moving them into real-world applications so that they ultimately help improve health outcomes in the real world. And finally, the for everyone piece indicates that we're not just interested in new types of medical breakthroughs. We're also interested in efforts that improve the access and the affordability of interventions that already exist, so that way, at the end of the day, we can make sure that today's best-in-class interventions reach the patient populations that need them the most. So ARPA-H is unique in that it has a number of authorities and structures that allow it to function very nimbly and quickly. So we are an independent agency, research funding agency inside of HHS and associated with NIH. We are an external funding agency, and we focused on contracts instead of grants and cooperative agreements instead of grants so that we can fund outcomes-based awards. We have a very lean and nimble management structure where we hire program managers who come in with ideas about revolutionary new approaches that we might take to improve health outcomes. And our director reports directly to the Secretary of HHS with a dotted line to the NIH director. Program managers come in for a relatively short period of time, 3 to 6 years, with an idea of both an important problem space, a deep understanding of the limitations of the current state of the art, and also ideas about a new way of approaching important problems. And the program managers, as folks that come in with these revolutionary new ideas, they shape the nature of the portfolio and what we invest in. And by having folks that are term limited, that helps us as an organization stay nimble and stay at the scientific forefront, because we are constantly pulling in top talent in emerging technology areas. And then our decision-making processes are very bottoms up, where the program managers have the autonomy to manage research portfolios in a way that gets us to those positive outcomes that we are interested in. So we, as ARPA-H, we are still a relatively new organization. I think today is our second birthday. So we've been around for just 2 years now, and we're continuing to build our network of organizations that both encompass our customers, so those who will ultimately make use of the technologies and new therapies and new treatments that come out of ARPA-H programs, so our patients, healthcare providers, and the public. And then our performer base. So these are the folks that perform the research that moves toward those large revolutionary program goals. And folks can come from many different groups, academia, industry, including small and large businesses, NGOs, nonprofits, and patient groups, hospital systems, et cetera. So we are very interested in continuing to expand the groups of organizations that help us work toward the research goals. And finally, we're building our network of stakeholders, both in the public and the private sector, who ultimately will assist with the long-term sustainment. So our programs tend to be relatively short in time duration, often 3 to 5 years. And we partner with other organizations to scale and expand and sustain the work coming out of ARPA-H programs in the long term. We also have a number of partnerships on the regulatory side with organizations like FDA and CMS, so that as we are setting up new programs and new research thrusts, we're also talking with important partners about the regulatory pathway and the potential pathways toward payments. So we can have a strategy for ensuring that technologies and new types of treatments that are developed through an ARPA-H program have a pathway toward that long-term sustainment and funding. ARPA-H organization has 5 different offices, 4 of which focus on these large revolutionary programs that are really pushing the envelope on the R&D side, and then a 5th office that focuses specifically on transition, which is that piece where we're looking for that long-term sustainment and the ability for the technologies to survive in the wild. So first, we have our Health Science Futures program, and this one's working to expand what's technically possible. So it's looking to accelerate advancements in the biomedical space and change our perception of what's possible, looking at things like platform technologies that can help us treat many different diseases at the same time. Then we have our Scalable Solutions Office, which is about access and affordability. So this office specifically focuses on these areas where maybe we have an intervention that works, but it's not necessarily reaching the patient population that needs it. So for instance, perhaps we have a certain type of technology that's only available in an academic medical center, and we want to miniaturize it so that it can move into a home health setting. Or perhaps we have some type of therapy that's costing $2 million per dose. We want to invest in advanced manufacturing in order to bring that price point down so that therapy is available to all of the patients that would benefit from it. And third, we have our Proactive Health Office, which is interested in prolonging periods of health and well-being. So this gets into things like preventative care or early detection and other types of technologies that might help Americans extend their periods of health and well-being and perhaps maintain their autonomy as they age in place. Finally, we have the Resilient Systems Office. So I'm the director of the Resilient Systems Office, and Andrea will be announcing one of the programs inside of our office. We're interested in building a more integrated healthcare system. So looking holistically at the combination of the health ecosystem, both from a technology and a people perspective. So the combination of the technology and people together, and looking at how we can make systemic changes in order to make the health ecosystem more adaptable, more resilient, and better able to continue functioning well, even if there's some type of unexpected disruption, such as a pandemic or a cyber attack or a disruption in our supply chains. And ultimately, we'd like to get to the point where we can anticipate and mitigate potential disruptions before most people notice that they're even there. And then finally, our Project Accelerator Transition Office is looking at those partnerships on the regulatory side or partnerships with venture capital or other types of organizations that can help take the technologies coming out of the first four offices and help create a path toward long-term sustainment in the real world. So talking for a moment about the Resilient Systems Office, these are the thrust areas inside of that office. So we're particularly interested in socio-technical system innovation, so ways in which we can adapt our technologies to better support patients, providers, and communities so that the healthcare is easier to access, delivered in a way that supports high quality outcomes and really supports the need of both our clinicians and our patients. We also have a subthrust on health ecosystem integrations, so how we can look at strengthening connectivity across the systems that support both our health and our public health systems. So this can include things like data interoperability, can also look at the way in which the ecosystem is moving from a technology perspective. For instance, by integrating artificial intelligence in new ways, and we're particularly interested in looking at things like the safety, security, and privacy considerations and technical advances that can help those innovations be useful and continue to be a value add for patients and clinicians. And then finally, the last area is looking at adaptive and anti-fragile solutions. So this gets into areas where perhaps there's an unexpected event. Are there ways that we can either mitigate or create a more adaptive way of thinking about how we structure the health ecosystem? So for instance, we do have a solicitation out right now for a program called Paradigm that's looking into distributed care platforms. So ways in which we can take hospital-grade care and move it out into a mobile setting so that it can reach every rural county in America. So we're interested in that type of way to add adaptability to our health ecosystem, and also ways to better anticipate and mitigate and respond to unexpected disruptions. So ARPA-H does have a number of programs that have launched to date. So we are doing things in areas from antimicrobial resistance to cybersecurity to these distributed health platforms, curing osteoarthritis, and looking to enable eye transplants where the eyes where we can reconnect the optic nerve in order to restore sight. So our programs will continue to be published, so please check back with the ARPA-H website at a regular basis to learn what we are doing next. So in terms of the way that we're structured, again, it's a very bottoms-up type organization where ideas for what we invest in are coming from our program managers who stay for 3 to 5 years, or stay for 3 to 6 years, and develop a series of programs that on average last about 3 to 5. So the program managers will come in with these revolutionary ideas. So again, a deep understanding of a problem space, the limitations of the current state of the art, and a novel approach that just might work in order to attract that problem area in a different way. And then we'll empower our program managers to obtain resources in order to test out their ideas at a scale where we can really tell whether that concept will work. So the program managers will come into ARPA-H and first start out by designing programs using what we call our Hallmeyer framework. So this is a series of questions that help to do just that piece of identifying that problem, limitations of the current state of the art, and our what exactly makes the approach new and revolutionary. There's a bit more to it where we're also looking to understand how it relates to what is really cutting edge, right? So are there reasons to believe that the problem that the program manager seeks to solve is feasible? Is there a reason to believe that within the next 3 to 5 years, we could make significant progress? And then the program manager will put out a solicitation, just like in ADAPT, which you'll hear about today, select a performer team. And then with that performer community, we'll be working to obtain the research goals of that program. So throughout the program, we'll execute and measure and evaluate whether certain milestones are being met in order to really move the technology concept forward. We're collecting lessons learned, both in terms of adapting what's happening inside of a program to better meet its goals, and also informing where we might want to invest next. And ultimately, we're interested in that commercialization and transition in order to ultimately create the long-term sustainment for each of our programs. And with that, I would love to reintroduce Andrea to talk to us about the ADAPT program.
Thank you so much, Dr. Roberts, for the overview of ARPA-H. And thank you, everyone, for coming to hear more about ADAPT. Again, my name is Andrea Bild. I'm the program manager for ADAPT, which stands for Advanced Analysis for Precision Cancer Therapy. This program asks, what if we could adapt cancer treatments as tumors mutate and change? The ADAPT program will develop adaptive cancer treatment techniques that detect when and how tumors evolve to a drug-resistant state, develop new, more accurate predictive biomarkers to recommend updates to a patient's treatment plan, and evaluate patient response to a flexible clinical trial design. ADAPT will target metastatic cancer, which causes approximately 20% of deaths in the US and affects over 600,000 Americans. Metastatic cancer can develop resistance to therapy quickly, highlighting the need for a systematic approach to solve this critical medical problem. There's also a large economic burden of costs from cancer care in the US, over $200 billion in 2020, with these costs rising. ADAPT will revolutionize how we treat metastatic cancer patients by identifying and targeting resistant tumor traits, thereby matching the right drug to each tumor as it evolves. We hope our goal is to more effectively have inhibition of tumor growth and increase patient survival time. Research has shown that cancer mutates and evolves quickly. ADAPT will identify and target these changes. Shown on this slide is a simplified diagram of a heterogeneous tumor where different colored cells represent different traits that have different responses to therapy. In the top panel, a tumor is depicted with cells that have two different genetic and phenotypic traits, pink and light blue. For example, this could be a breast tumor where the pink cells are positive for estrogen receptor expression and responsive to endocrine therapy, while the light blue cells lack estrogen receptor expression and are resistant to endocrine therapy. Similarly, this could be a lung tumor where the pink cells have an EGFR mutation and respond to therapy targeting this receptor, while the light blue cells do not have this mutation and do not respond to this therapy. So patients in this depiction are given the pink therapy, and the pink therapy or the pink cells respond while the blue cells do not, as they have different traits that enable them to continue growing during treatment. Now, because the tumor measurements aren't always taken frequently, the increase in the number of blue cells is not detected, and the patient continues to be treated with this initial pink therapy because the tumor's overall size decreases. But the light blue resistant cells continue to grow and also evolve and mutate into the dark blue cells. As the tumor grows larger, doctors will change to a different therapy, in this case, the green therapy. But as this treatment is not based on a tumor's evolution and current resistant traits, the tumor continues to grow and evolve. Thus, in the current state, we know that cancer changes during treatment, but therapies are not often informed by tumor changes or tailored to acquired resistant traits, as there is only limited tumor measurements and biomarker analyses, which are most often performed prior to treatment. The ADAPT approach is shown in the bottom panel. Here, while the cancer patient may start with the same therapy based on pre-treatment classification, more frequent tumor measurements show that the light blue cells continue to grow during the pink treatment. Therefore, a light blue treatment is added to target these cells. Now, as this is metastatic cancer, the cells continue to evolve and change into the dark blue cells. But with additional tumor measurements, we can again adapt treatment to target these changes with the dark blue treatment. In this way, we aim to improve patient response to treatment and outcomes. So in ADAPT, therapies will be informed by and tailored to the observed clinical resistant traits using in-depth tumor measurements and biomarkers taken before, during, and after treatment. So ADAPT's predictive biomarkers will identify the right therapy for the right patients at the right time. As shown on the left, many traditional cancer biomarker approaches have focused on single gene mutations and single data types, such as pathological measurements, often taken from biopsies prior to treatment. Relying on a narrow range of data or genes and limited time points may not fully reflect the complexity of a tumor signaling and could miss detecting and acting on emergent resistant traits. As shown on the right, the ADAPT program will collect, integrate, and analyze a diversity of data types with advanced computational methods to build new, more accurate, predictive, multimodal, multi-gene biomarkers for individualized therapy strategy. So our goals for this program are to increase the number of cancer treatments that are informed by multimodal biomarkers based on multiple genes from approximately 5% now to 100%, and to increase the percent of metastatic patients that respond to standard therapies after initial treatment as we're personalizing care for these patients. The foundation of this work will be a clinical trial framework that allows us to identify, test, and validate predictive multimodal biomarkers and inform patient treatments. So there are three technical areas which we refer to as TAs in ADAPT. TA1 is focused on therapy recommendation techniques. TA 1.1 performers will apply multimodal data fusion methods to integrate diverse data types for TA 1.2, which covers resistant trait modeling during tumor evolution, and TA 1.3, which builds biomarkers that predict drug response. TA 2 in the middle is the evolutionary clinical trial. For TA 2.1, the ADAPT program will collect a diversity of tumor biology and clinical measurements during 2.2, which is the centralized modular clinical trial protocol with multiple sequences of different therapies to support the rapid development and testing of predictive biomarkers in TA 2.3. And then lastly, on the right, for TA 3, the ADAPT program will unify insights from researchers and clinicians through the creation of a shared treatment and analysis platform. This open source platform will enable rapid availability of integrated patient data, clinical trial protocols, and the ability to design, test, and standardize newly developed biomarkers. Through this platform, a close community of researchers and clinicians and their collective knowledge base will be formed to facilitate translational oncology research. Next, I will take you through the adaptive cancer treatment platform. The top part of this figure represents the therapy recommendation techniques. Shown here, tumor measurements are used to analyze tumor biology, such as the pink and blue cells traits prior to treatment, and discover new predictive biomarkers for multimodal data that ultimately aims to recommend optimal therapy. Then, for TA2, the evolutionary clinical trial on the bottom part of the figure will iteratively enable clinicians to dynamically adapt therapy based on real near-time changes in tumor biology. In addition, new tumor measurements are taken after treatment or when patients progressed and analyzed by the TA1 teams to identify the acquired resistant traits in new biomarkers. These tumor measurements and biomarker algorithms are shared in our TA3 treatment and analysis platform, which is the collaboration space that accelerates the development and sharing of data models and trial protocols. New tumor biology, such as the growing blue cells shown here, and biomarkers can then be discovered to identify the next line blue therapy based on the acquired traits during tumor evolution. The iterative nature of this program and the tight collaboration across research and clinical teams enables targeting tumor evolution through multiple lines of therapy. This cycle develops a new approach in the treatment of cancer patients where data rapidly, consistently, and accurately informs patient care. So shown here is just a summary of the advances to the current state of the art, which includes a development of methods and data to track cancer resistance and identify personalized therapies at the speed of relevance. Also, to develop, test, and apply multimodal, multi-gene drug response predictive biomarkers within a clinical trial infrastructure in near real time. Thirdly, to develop modular evolutionary protocols, enabling the study of multiple cancer types and treatment lines. And then lastly, an infrastructure for researchers and clinicians to work together, share data and protocols, and carry out integrated translational studies. I'll now review in more detail each of the three TAs. Shown here first is TA1, the therapy recommendation techniques. As shown in the top panel, many current clinical trials do not include multiple lines of treatment or measure tumor traits during and after therapy. This misses opportunities for this information to help inform the next treatment line as new resistant traits develop. On the bottom, ADAPT will address this need by collecting deep data during multiple sequential lines of therapy with deep tumor measurements that allow for identification and targeting resistant traits through biomarker development. As patients will be followed during different treatment regimens and different cancer types, it's important that TA1 biomarker teams develop an approach that can build multiple biomarkers from multiple lines of therapy. Shown here are the three sub-TAs. TA 1.1 will develop methods to fuse multimodal data and generate a body of high quality, complex, and integrated data, which will enhance the downstream resistant trait and biomarker analysis conducted by TA 1.2 and TA 1.3 performers. TA 1.1 performers will include teams that are skilled in early fusion methods that integrate data types prior to downstream analysis, as well as late fusion methods that integrate biomarkers from individual data types into one single predictor. It is safe to assume here that many of the tumor biology measurements will be blood-based, such as circulating tumor DNA, RNA, methylome, and clinical trial data such as blood counts and other data types, as well as upfront bulk RNA, DNA-seq, single-cell analysis, in some cases, post-treatment tumor measurements. For TA 1.2, which is the resistant trait modeling, ADAPT will expand our knowledge on tumor evolution by analyzing real-world longitudinal biology data to discover therapy-resistant traits throughout the course of treatment in the evolutionary clinical trial. These teams will develop sophisticated models that trace tumor-resistant patterns, fostering the ongoing identification of innovative drug targets and regimens that are more effective and precisely treat the unique tumor traits found in each patient. Performers for this TA can include those that have both computational and wet lab components to study the resistant traits that are required during tumor evolution. And lastly, on the right, the predictive biomarker teams in TA 1.3 will develop a computational approach to build multimodal biomarkers across multiple lines of therapy and across different cancer types. Performers will use longitudinal data to develop new predictive biomarkers and allow for iterative development, testing, and validation of these biomarkers as part of adaptive treatment regimens. Importantly, TA 1.3 performers will build biomarkers that can be tested side by side concurrently in the clinical trial for a more efficient biomarker development and testing process. We will no longer need one trial to separately test each biomarker. For TA1, applicants can focus on one, two, or all of the sub-TAs. It is very important to meet the specifications listed in the module announcement, so please consider the module announcement online as the ground truth. TA 1.1 applications must address either early data fusion, late data fusion, or both in a reproducible manner. Performers or applicants for TA 1.2 for resistant trait modeling must analyze the diversity of data types being collected and must identify pathways, genes, and proteins that emerge in acquired resistant mechanisms during cancer progression. These approaches must include dynamical temporal modeling, so they can't focus on single individual time points, and they must be reproducible and transparent. If you have a black box method that you want to use, this is not the right program for you. Lastly, for TA 1.3, the biomarker developers, these performers will use advanced mathematical computational approaches for prediction of drug response, So we're looking for innovative approaches, innovative, sophisticated, that can deal with the complexity of the data that will be analyzed. Again, you must use multimodal data types to build these biomarkers. And the biomarkers must use a complexity of data for predictions. So a minimum of 5 mutations, transcripts, or proteins, hopefully more, and hopefully biology based. And again, approaches for TA 1.3 also have to be reproducible, transparent, and testable by external reviewers. In addition, for TA 1.3, existing biomarkers previously applied to patient data that have been demonstrated are welcome, but a general pipeline should be proposed for biomarker development as well, and interpretable models that are informed by tumor biology and signaling are preferred. We're at ARPA-H, so we have aggressive metrics. So again, refer to the module announcement for further details on metrics, but in your application, please discuss how you will meet these metrics at each stage. So for the ADAPT program, there are 3 independent stages: phase 1, which has a stage 1, which is the first 6 months, and phase 2, which is 7 months leading up to 3 years, and then phase 2, which is the 4th through 6th year. I won't go through all of these in detail. It's just here to remind you to study the metrics from the module announcement and include them in your application. I'll just go through one. So for example, you can see here for TA 1.3, we have a component where therapies must have an associated multimodal biomarker. The ground truth at phase 1 is 5% of therapies might have these types of biomarkers. But by the end of 3 years, we hope that in the clinical trial therapies that are selected, over 30% have a multimodal biomarker. And then by the end of the program, we have over 90%. So please, again, pay close attention to the metrics in the announcement when drafting your program. Next is TA2, which is the evolutionary clinical trial, with on the left showing the current state and on the right showing the future state. And if you look to the bottom, there are many types of clinical trials currently available. And shown here is just one of them on the bottom, where all patients are randomized to either one of 2 treatments, treatment 1 and treatment 2. And at the end of the clinical trial, you get an understanding of which treatment works best in that particular population. In many of these trials, but not all, multimodal genetic and molecular analysis is not performed, which does prevent tailoring of therapies to an individual specific tumor. So this is population level. Also, because only one sequence of treatment is tested in a clinical trial, this misses out on measuring and targeting evolutionary resistance over multiple lines of treatment. So ideally, you'd understand why some patients are no longer responding to therapy, what the acquired resistant mechanisms are, and then targeting those in the next line therapy. So on the right shows the adaptive evolutionary clinical trial, where you have 2 patients where therapy is personalized to their particular tumor. Patient A is given the red treatment to target the red cells. Patient B is given the green treatment to target the green cells. So in this trial, we hope to assign patients to a specific treatment based on tumor biology measurements and to individualize their particular treatments that they receive. In the ADAPT evolutionary trial, we'll follow patients under one protocol as tumor change during multiple lines of treatment and individual treatments for each of these time points. Our expectation is by personalizing therapy over time, we can acquire higher drug response rates. So proposals for TA2 must address all sub-TAs, TA 2.1 through 3. For TA 2.1, serial tumor measurements are required to understand how a patient's unique cancer changes in response to treatment. Tumor biology measurement technologies include blood-based circulating tumor measurements, single-cell sequencing, bulk tumor omics, and spatial imaging, as well as others that can be collected serially before, during, and after treatment, and combined with clinical data such as electronic health records, imaging, and pathology data to inform biomarker development. These tumor tissue, blood-based, and clinical data will be collected more frequently than standard clinical trials, enabling patient tumor data to inform treatment. For applicants, we will coordinate collection of diverse data types. Thus, if, for example, you don't have a collaborator with methylation analysis, please don't worry, as we will help organize collection and measurement types across the program. In the middle, the meat of TA2 is TA 2.2, which is the new modular trial protocol that integrates existing or new approaches, such as adaptive trials, randomized trials, and one trials, and that flexibly adapts treatment arms over time for each cancer subtype and drug treatment. This flexible and agile approach of the ADAPT clinical trial framework is intended to maximize the likelihood of identifying effective treatments for a variety of patient subgroups as they remain under a single protocol and as tumors and treatments change. I understand this is a challenging TA and are looking forward to your best and most creative ideas. TA 2.2 trials must be focused on metastatic cancer patients and include multiple lines of treatments that would benefit from biomarkers for treatment decisions. TA2 performers are not required to include biomarkers, as TA1 performers will build these biomarkers, although applicants can apply for both TA1 and 2. Again, please pay special attention to the details in the module announcement. And lastly, on the right for TA 2.3, biomarkers developed by TA1 performers will be integrated into the evolutionary clinical trial for testing and validation, thereby creating a rapid feedback cycle between research and clinical practice. Oftentimes, cancer biomarkers are not translated into clinical care, as it's challenging for each individual researcher to carry out their own clinical trial. By incorporating biomarker discovery and validation directly into the clinical trial process, we aim to increase translation and speeded discovery and implementation. ADAPT will include a diversity of cancer types to build robust biomarkers and generalizable methods across diverse patient populations and treatments. ADAPT aims to initially focus on breast, lung, and colon cancer, which together comprise almost 40% of cancer deaths in the US, with colon cancer an emergent global epidemic among people younger than 15. However, if you are interested in proposing other cancer types, I highly recommend that you submit a solution summary, which is a 3-page document we will detail later on for consideration. Again, TA2 specifications are listed here, or some of them are listed here. All of them are in the module announcement. For tumor measurement technologies, we just require a diversity of tumor biology data types to be collected of sufficient mass and quality to support research. And again, because we're moving fast, analysis and measurements must be feasible within 1 month from collection and processing. For the clinical trial requirements, again, this program is focused on metastatic cancer and includes multiple lines of therapy under a single protocol with a flexible approach that allows changes in design. We require serial tumor blood-based measurements and because we have limited time for the ADAPT program, please pay close attention to the progression-free survival average of your different treatment lines. And lastly, the protocol must include a mechanism for biomarker evaluation during the multiple lines of treatment. I also wanted to say that additional details in the application to address are listed in the module announcement. This includes things such as the clinical trial design, the treatment regimens, plans for biomarker testing, the ability to collect clinical data, your historical clinical trial enrollment, and patient demographic and diversity measurements. This is just a ping to remind you to look at the module announcement for the application. Again, for the metrics, we are taking some aggressive kind of metrics here. Again, for this first stage, which is the first 6 months, we expect that time will be spent getting approval for the protocol and beginning to open the sites. And then for the next 3 years, increasing sites opening with patient enrollment from approximately 200 to 500, with by the end of 6 years having all of your sites open and up to 1,000 patients enrolled. There are other metrics to include in your proposal. Please pay close attention to all of these, even though I'm only highlighting one, to make sure that you can address them in your proposal. So for TA3, the ADAPT program seeks to enable dynamic collaboration between researchers and clinicians to guide treatment. TA3 will create a collaborative platform and testing ground that bridges the research and clinical domains. The platform's de-identified research and clinical data algorithms and clinical trial protocols will be made publicly available through the ADAPT portal, thus creating a new standard for the field and alleviating roadblocks that have limited the impact of big data in basic and translational cancer research. Some of the key features of the treatment analysis platform include data harmonization, architecture, and use. Performers will improve the volume and insight richness of cancer data through data annotation, standardization, and harmonization, and ensuring compatibility, consistency across data types. Performers will also develop an architecture for cancer data algorithms and pipelines through a data lake and accessible data warehouse. And integrated data will be utilized in the platform to enable near real-time data availability. Lastly, performers will develop a user-friendly platform that enables clinicians, researchers, and others to design, analyze, and share innovations from TA1 and 2. Again, TA3 has specifications that are listed here, which include a scalable data management solution that stores the raw and processed data that it's collected, a computational pipeline to rapidly process tumor biology measurements and perform other functions, implementation plan for data harmonization, and a development of open APIs. The metrics for TA3 are shown here. And for TA3 in particular, we have a metric that is pretty robust, that you must have your portal up and running and available to engage with the performers and community to share data pretty rapidly. So again, please pay close attention to these metrics so that you can meet them by the phase 1 and phase 2. At ARPA-H, there's an emphasis for accelerating innovation and discovery. So we have put together an ambitious timeline for transforming translational research in clinical trials. This timeline reflects our goals of identifying new resistant traits and developing a multitude of predictive biomarkers for FDA submission and enrolling up to 1,000 patients with resulting improvements in PFS, progression-free survival, as well as building the data analysis platform for research efforts. We're aiming to launch our first clinical trials in early 2025, 6 months after the program starts, followed by quick discovery of resistance mechanisms and biomarkers in early 2026, and sharing of de-identified data with the public starting mid-2026. We have some ambitious goals and key outcomes for the program listed here. For TA1, the therapy recommendation techniques, for the first time, a consistent mechanism will exist to build and clinically validate biomarkers for both standard of care and new therapies within the framework, the ADAPT program framework. Our goal is to have over 15 new multimodal biomarkers so that over 90% of standard of care therapies have an associated biomarker, as well as additional biomarkers on more targeted therapies. In the middle, for TA2, the evolutionary clinical trial, we'll have a novel clinical trial designed to implement biomarkers and treatment regimens targeting resistance and decreasing cancer growth, leading to longer patient survival time. Our goal for this is to have over 50% improvement in progression-free survival time across multiple treatment arms. We know this won't be all treatment arms, but we hope that some of them have this improvement. And then lastly, for TA3, for the cancer treatment analysis platform, we create an accessible data library and digital foundry for developing new therapeutic approaches in clinical trials. We expect that over 95% of the relevant clinical trial data will be available to the research and clinical communities for their use. ADAPT will impact multiple different shareholders, including patients, which we hope to benefit from improved care and personalization of their treatments through the biomarker development and application. The scientists, we hope that they can benefit from the clinical trial infrastructure and deep data to enable translational research discoveries. For the clinician, we hope to benefit these professionals to give better informed treatment decisions and drug selection for individualization of therapy. And then lastly, for insurers and pharmaceutical companies, we hope to, by identifying the right therapy to avoid unnecessary side effects from drugs that don't work, avoid treating patients with ineffective treatments, and identification of new cancer targets through the longitudinal data. It's important to note that for ADAPT and our ARPA-H programs, we want to make sure that we have a diversity in the programs. So the ADAPT evolutionary clinical trial will ensure equitable access for patients in the U.S. by establishing sites with high diversity. And for the clinical trials, the enrollment must match the cancer population that is being studied. Please, again, pay close attention in this module announcement for details. Okay, so next steps. The first next step is to submit a solution summary. We just posted on the website a special notice, so you can find this information on the website. And a solution summary is basically just an abstract submitted that you will get feedback on whether or not it's recommended or not recommended for a full proposal submission. While not required, it is highly recommended. We have over 500 people that are interested in the program, and this will save a lot of people time if your ideas are not in line with the ADAPT program. So I cannot emphasize enough to submit the solution summary within 2 weeks. So they're due March 29th, and the time is actually 10 o'clock a.m. So please submit by 10 o'clock a.m. on March 29th, Eastern Standard Time. And this gives us the opportunity to review those quickly over the weekend and the following week. The portal to submit the solution summary is shown here. And again, this is a highly recommended step if you're interested in the ADAPT program. Also, there are many of you that might want to team with other groups to build your program. And there is a teaming page available online at the ARPA-H site and also detailed here. You can input your information and you'll be able to see others' information, which will allow you to team up with other groups that might be complementary to the research that you want to carry out. And then also lastly, we just wanted to give you links to all the resources that you might need for your application. Those are shown here. We have the ADAPT program page, which again, you can find the ground truth of all information. And if you have questions, please submit them to us and they will be posted. The answers will be posted online for you so that you can review these as you're preparing your submission. All right. So with that, we're done with the summary of the ADAPT program. I was so excited to present this to you. So thank you so much for coming today. Next is more information on the submission process by Caitlin Burns from the Business Innovation Division. Take it away, Caitlin.
Thank you, Andrea. So we're going to do a little bit of a pivot now from the typically more exciting technical side of the house over to the administrative side of the house. And this is what ensures that all the solutions you have and everything get the attention they deserve from the Business Innovation Division. And it allows us to streamline the process and really kind of make sure that your proposals are in line with what we are hoping for. So let's go to the next slide, please. This is kind of an overview. So the Master Announcement Instructions, also known as the MAI, might be new to a lot of you. So the MAI is kind of your top level overview. So everything in the MAI will flow down to the module announcement. The module announcement has the program specifics, all the technical details, everything that applies specifically to this ADAPT program, but you'll want to use them in conjunction. So while you're kind of creating your solutions, you'll want to make sure that you are referring back to the upper level MAI instructions and using that as your manual as you go through the process. Next slide, please. So these are just some kind of overviews, notable dates. Obviously, we're here at the Proposers Day. Please note the due date for questions and answers. And then again, as Andrea mentioned, the solution summary is a new addition. So please note the due date there. As outlined in the solicitation, this is the program schedule. It's a 6-year effort. There's 2 phases. And one of the things that we really wanted to highlight was the open source standards. So we have an emphasis there. If you have any questions, please submit those. We'd be happy to answer them to make sure that your thoughts and your proposal are aligning with what we are hoping to see there. And same with the software component standards. So we have language in the solicitation that should help you, but these are kind of 2 focus areas. So please submit any questions and ensure that you are aligning your solution with our preferred vision there. So next slide, please. This is a screenshot from the solicitation, so hopefully you have seen this before. It kind of breaks down more the module categories and how we have structured this. Pay attention specifically to the individual versus the combined proposals. There's a little bit of nuance there. And we just wanted to foot stomp again that while these have dollar value brackets, we would like a very thoughtful proposal, thoughtful analysis on your end, not just automatically going to the top of any of the brackets. That helps us help you streamlining the process and make sure that we don't need to ask for more information than necessary in the next step to kind of get to our justification there. So just want to foot stomp that we would really, really appreciate thoughtful analysis there. Okay, so this is just kind of more basics. And stage 1, this is what we're requesting. Again, this is all spelled out in the solicitation. If selected, then each proposer will move to the stage 2, volume 2, where we'll want a little more detail to kind of get to that next level. There's a bundle of attachments for each stage that are we would like to be submitted. Stage 2 bundle of attachments will be provided to those who have been selected to move to that stage. So right now the focus should be on the bundle 1 attachments that are out with the solicitation. An overview of OT. So here at ARPA-H, I believe is Dr. Roberts who kind of touched on this. OTs are really kind of our bread and butter right now. They are an agreement, so they have kind of the elements of what your typical contract would have with, signature, everything like that, but they're designed to reflect more commercial practices than the traditional FAR practices that can be rather restrictive and difficult to work with for what we consider non-traditional vendors. The biggest thing here is they really are collaborative. It's a partnership between ARPA-H and the proposer. We want to structure it as the cliche win-win. It's nobody trying to gain an advantage or anything like that, which allows for the more free-flowing negotiation because we don't have those restrictions. So we're really trying to find what works best with both sides, and OTs allow us the freedom to work in that space. Again, just some pros and cons. So pros, there's a lot of laws and regulations that do not apply. We are allowed to follow more commercial standards that a lot of proposers are probably used to, and it can very much streamline the award process. With that said, on the con side, it can take longer to negotiate because we're trying to come to that very sweet spot in the middle of what works best for each side. So with fewer guardrails, that can lead to some hesitation on those that are new to working with the government, but we are very intentional and thoughtful in our negotiation process. And again, it's very much a two-sided effort and not a us versus them type of situation. Just a little bit of an overview. Obviously today, Proposers Day. If you are hearing this live, then you have attended. If not, it is not mandatory to have attended to submit anything. The solution summary, again, not mandatory, highly, highly, highly recommended. And that's really to help us help you. We don't want anybody spending their time, money, effort, putting together a full proposal if it's just not meeting the mark or not something that might be viable. For full proposals, again, going to foot stomp that number 3 bullet there. Please be thoughtful in your proposal, your dollar value, and your solution. And then the last bullet as well. So this is outlined in the solicitation. If you have questions on this, please submit them. There will be GFR provided, so that is not needed to be included in your proposal. The evaluation and selection, again, stage 2 will only be requested for those. It will not be open to everybody. So those that have potential through the stage 1 will be notified, and stage 2 information will be sent out at that point. These are the evaluation criteria. There's, again, it's outlined in the solicitation. I'm not going to foot stomp this here. I assume everybody has read that. So scientific and technical merit, capabilities, experience, contribution to the mission. And if you go to the next slide, the price and value analysis. So these are pretty standard evaluation criteria. Again, outlined pretty clearly in the solicitation. So default to that if needed. A few more things. So as you all have hopefully already seen, any changes or announcements, they will be posted. What is posted should always be your default. Anything said today, if there's a disagreement between what is said and what's posted, please let us know, but the posted information is the record. Again, please read the MAI, all module announcements. The MAI structure is probably new to a lot, so ensure that you understand what we're looking for there. because non-conforming proposals will not be evaluated. So ensure that you're in compliance with everything that we're asking for there. On the right-hand side, the dates and deadlines, again, that's 10 a.m. So please make a note of that. And that probably underlines my plea that you default to what is posted and not necessarily what is here, as we are human and prone to mistakes. So if you see anything like that or notice anything else, please raise it to our attention so we can ensure that we have it corrected, but the posted information is the default. There's the link to submit your questions if you have anything else, either from today or anything that might be raised. Please submit them there and we will get those answers out. Just a few important reminders. Again, foot stomping solicitation is your ultimate reference. The number of proposals, so please read through that. Again, like the budget limits, everything are for the total cost. If you have any questions there, something isn't clear after reading the MAI and everything posted, please reach out. We can clarify that. And just a little bit of information for any international entities. Again, here's an awesome, you can just pull this up on your phone, ask a question if you have it, and we will get answers out as soon as we are able. And I believe that is my last slide. So hopefully the administrative side of the house didn't put anybody to sleep, and you can get back to focusing on the technical side. Thank you.
Thank you so much, Caitlin. I just want to also note here that a PI might submit one proposal as a prime, but you can be a sub-award on multiple projects. So if you're the PI, you can only submit one as PI, but you can be on other proposals as a sub-award. There is a recording available online if you need to review any of this information. Again, everything is also posted online if you need to refer to it. We just wanted to end with just some important reminders about technical requirements. Again, for the cancer types addressed by ADAPT, we're initially looking to focus on metastatic breast, lung, or colon cancers. However, if you have an innovative solution for another cancer type, please send it in an abstract, also known as a solution summary, by the March 29th date. For previously developed biomarkers, you can include existing biomarkers previously applied to patient data and that have demonstrated significant predictive capability. However, you must also address the development of a pipeline for developing biomarkers across different treatments and cancer types. We also want to state that we are working closely with the FDA. Performers who are selected for the program will subsequently work with our ARPA-H team and an FDA team for insights into the regulatory science supporting the evaluation of new technologies. So if you come on board as a performer, we are already working with the FDA, and that will be part of the program that you'll be working with. There are also important technical requirements on the capabilities of proposals in teaming. Proposals to TA2 do not need to address biomarker development capabilities as long as the clinical trial includes treatments that would benefit from biomarkers. Teams with clinical capabilities who wish to team with biomarker developers, again, use the teaming page if you would like. Proposals to TA1 need not address clinical capabilities. So TA 1.3 applicants can develop computational pipelines for biomarker development across different therapies with multimodal data, even if you aren't currently linked to a clinical trial. Just interested in your ideas and how you will build these biomarkers and apply them more broadly. We will then link you to the TA2 teams in the clinical trial. You can, again, also team up with the clinical trial folks on the teaming page. For research organizations or CROs, the ADAPT team will select a CRO to manage the centralized clinical trial components and data collection. So TA2 proposals should not include CRO costs in their budgets, nor should they include data storage and analysis costs, as these will be covered by the ADAPT program. CROs should not apply to ADAPT. This will be a direct relationship with the ADAPT program. I guess that's all. Thank you everyone for coming. We are so excited to work with you on ADAPT. Thank you for your interest. Please refer to all the online information, and we look forward to seeing your great ideas. Thank you so much.